It's great to be back in Brazil, and I was here just a few weeks ago and attending a really interesting set of talks which were given entirely in Portuguese. And so as I was listening to Shelby talk about the difference between heterozygosity and homozygosity in the MHC, it made me think of the advantages of bilingualism, and there have been some interesting studies published. So for those of you who are sitting and listening in English, which is not your mother language, I think that your brain cells are being stimulated, you're going to live much longer, you're going to avoid dementia. It's fantastic. So congratulations, because I know it is a chore to listen in a foreign language. And I apologize that my Portuguese is not good enough to give the lecture in Portuguese. I realized this in a taxi last night, where I thought I had said one number, and the taxi driver thought I had said another number, and those numbers were a long way apart, and so it took a while of negotiations to get back. So I'm going to talk to you today about two different areas. One I'm going to talk about is junk DNA, and since it's after lunch, I feel I can show a clinical picture, and although it may not project terribly well, these are slides of someone who is suffering from an autoimmune skin disease called psoriasis. And for those of you who would like to know what I'm going to tell you in advance, I'm going to show you data that HIV infection stimulates, transcribes, and we have translation of some of the fossil ancient viruses, the endogenous retroviruses that are within our DNA, and we think that this actually might provide an opportunity to attack HIV in a different way through vaccination or therapeutics. And I'm going to show you some data, both published and unpublished, on that. And then it's interesting, coming to a lot of meetings, you get to understand one phrase from one person, one from another. And I was at two meetings a while ago, and Rafik Sekali said one thing, and Hendrik Streek 1 week later said another thing. And put those two together, and we've been investigating people who are susceptible to the skin disease psoriasis, and we find, to our surprise, that the genetics of people who are susceptible to psoriasis are extremely similar to those that naturally control HIV. And you've heard about this from Dave Watkins and a bit from Shelby. But what that's led us to be able to do is to begin to dissect out why there are certain antiviral mechanisms. And I think it's hopefully going to be an interesting story for you. But at the beginning of the course, I'm sure people showed these type of maps. In the global scientific community we live, I'm over here in San Francisco and enjoy coming down here to Brazil. When you look at the map of the world by the number of HIV infections, you realize that every country is affected, and the great burden is in sub-Saharan Africa. So of course treatments are very important in helping to reduce morbidity and mortality. And I think the last 2 years have been really quite exciting in relation to the number of ways in which we can potentially prevent people from becoming infected with HIV. And Bob Grant's work and colleagues have really brought into play this exciting opportunity of treating people with antiretroviral drugs, those who are high risk from infection, and preventing people who take those pills from becoming infected. Passive antibody studies in non-human primates are also encouraging, and in the HIV vaccine field, I think all of us have been perplexed and excited at the same time about the results of the RV144 TIDE trial which has shown efficacy at a very modest level. And so there's a lot of information being drawn out of that. Nelson Michaels' paper is, I think, coming out next week in the New England Journal of Medicine, which hopefully will shed more lights on the correlates of protection. But I'm going to go back to these two things that we've been interested in relation to HIV pathogenesis, and I'm going to start talking about these fossil viruses. And this has been a very close collaboration with Mario Ostrovsky's lab at the University of Toronto and an ex-grad student of his, Brad Jones. So when I was doing my basic biochemistry, we sort of passed over what people would call junk DNA. And as you know, the gene coded within the DNA has both exons and introns, and when it's transcribed, those introns are spliced out and then translated into a more familiar amino acid sequence. So I think a lot of people just wrote these introns off as being not very interesting, not very relevant, probably not important. But when you look at the whole of the human genomic material, you see the fraction of the pie that codes for exons is really very small. And there's a large amount of repetitive DNA material that includes the long interspersed nuclear elements, ALU sequences, other types of repetitive DNA, introns and regulatory sequences. And so when we think about the genome, and now with sequencing of the genome, it's going to be interesting to see whether more emphasis is put into non-exonic material. There's really only a small fraction that is coded. So what about this junk DNA? Well, someone came into my office and took this picture. This is what life is like for scientists in the United States now, writing grants. The question that Brad Jones and Keith Garrison, a postdoc in my lab, asked was, could there be anything useful hidden in all that trash? And so what we realized is that within that DNA are these fossil viruses. They're thought to be remnants of ancient infectious retroviruses which endogenized and have got transmitted and have stayed. So for mothers in the audience, that you will pass your genetic material to your daughter, and those endogenous retroviruses will still be present. If we think about the familiar infectious type retrovirus, of which HIV is an example, you have structural and non-structural gene products flanked by long terminal repeats. And endogenous retroviruses, human endogenous retroviruses, can come in a variety of forms. There are ones that have similarities to infectious retroviruses, but may have stop codons or mutations that generally mean that these endogenous retroelements are not transcribed. There are other forms where the envelope is deleted, or even a solitary LTR can be an endogenous retrovirus. So the familiar life cycle of an infectious virus such as HIV is one of attachment, penetration, reverse transcription, integration into chromosomal DNA, and then when that cell is activated, expression of retroviral genes, new viruses are produced, pop out, released to complete the life cycle. And infections that are limited to somatic cells will only allow for a horizontal transmission. But if that infectious retrovirus could infect cells of the germlines, then there would be passage of that genetic material from mother to offspring. And providing that endogenous retrovirus doesn't result in a strong disadvantage to the host, then the endogenous retrovirus would become fixed. And there are many of these endogenous retroviruses which are fixed. And not just in humans, but also in rodents and all animals studied. So once we started looking into endogenous retroviruses, we realized that it's a big field. There are lots of different types of endogenous retroviruses, and they can be grouped into different classifications. We have the beta retroviruses, of which HERV-K is the one we've studied most, HERV-K HML2, thought to be the most recent insertion. But there are older HERVs as well that have more similarities to gamma retroviruses, and also some with similarities to spumaviruses. And these endogenous retroviruses form 8% of the entire genome. I mean, it's incredible to think that 8% of everyone's genome in this room is composed of endogenous retroviruses. And a recent study from John Coffin's group has helped to continue the classification of these, and there are probably at least 96 different HERV lineages within the human genome. And most of these HERVs are replication incompetent due to mutations, but there are some endogenous retroviruses which have the potential for being transcribed and translated in certain times of development. If we think a bit more about that retroviral phylogeny, HIV is a lentivirus is over here, and the endogenous retroviruses being spread out here are quite genetically distinct from HIV. So there's no obvious sequence homology between HIV and endogenous retroviruses. Now we know from various biological studies that in some circumstances endogenous retroviruses have been beneficial to the host. So this little mouse here has this product called FV1, which actually can restrict the entry of another retrovirus, the friend virus, into its cell. And it was found that that FV1 is in fact a product of a mouse endogenous retrovirus. And within humans, at certain stages of development, different HERV families can be expressed. And one particularly interesting one is the envelope protein of a HERV-W that actually allows the formation of the syncytial trophoblast and allows pregnancy to progress the way that it does within humans. So Brad Jones and Keith Garrison in the lab realized that HIV doesn't infect a cell in the absence of a retrovirus infection, because HIV is already infecting a cell which contains an endogenous retrovirus. And the work of a number of different investigators have highlighted important proteins such as the APOBEC family, which were not evolved to work against HIV, but we think are present and maintained within the genome to help in the containment of these endogenous retroviruses. So perhaps it is not such a leap to think that HIV, with its VIF protein, which takes APOBEC to the immunoproteasome, would have an maybe unintended consequence of not only allowing HIV to complete its life cycle, but also to release normal controls of endogenous retroviruses. So Brad and Keith went about to search for increased endogenous retrovirus expression within the plasma of HIV-infected people. And they developed some molecular tools to do this, and then looked between HIV-positive and HIV-negative subjects. And using a calibrate or HIV-negative subjects, the amount of HERV-K expression in the plasma was extremely low, but when they looked at HIV-positive subjects, in all cases, the transcription was really much higher. So you may be thinking, what has this got to do with HIV pathogenesis? Well, I think the exciting development for us was when we realized that we might be able to use this information to a potential advantage. And this was the time when people had been trying to design vaccines designed to stimulate HIV-specific cytotoxic T cells. And we know that the diversity of HIV is one of the major problems in developing a vaccine. And also, if you're thinking about developing a vaccine which stimulates HIV-specific CTL, you want to have those cytotoxic T cells that are going to recognize that infected cell before new variants are produced. So collectively, we developed this hypothesis that if you could develop or elicit a CTL response that could specifically recognize HIV-infected cells without recognizing HIV antigens, then you may be able to kill and attack an HIV-infected cell and not have to worry about the diversity of the virus. And so we sort of call this a no HIV vaccine concept. And so the model looks like this, that if you generate HERV-specific immunity, you can attack an HIV-infected cell through this static antigen, and it wouldn't matter which HIV antigen was present. And I'm going to show you data in the talk that supports this hypothesis. So in order to get there first, we asked the questions, was there any recognition in HIV-infected people of endogenous retrovirus epitopes? And as you'll remember, I showed you that HIV in the lentivirus family is really quite genetically distinct from these endogenous retroviruses. But Keith Garrison took the perspective of a T-cell receptor which recognizes short peptide fragments. And you can make some alignments between HIV sequences, represented here HHB2 and endogenous retroviruses. And in red, I know it's a hard color to see, but it may come out somewhat. You see that there are areas where there are some amino acids which are shared, and you can make alignments, at least for reverse transcriptase and part of GAG. And then from a T-cell receptor perspective, what is highlighted in yellow are epitopes that are known to be CTL epitopes that are present in HIV infection. So with that knowledge, we made a set of peptides that had both similarity and also dissimilarity in sequences. So if you look at this first peptide, this is the HIV sequence, this is the HERV sequence, and there are 6 amino acids shared between them. And we made a set of peptides and then asked the question, would HIV-infected people make responses to endogenous retroviruses? We initially looked in people with primary HIV infection, and we used a standard ELISpot assay to interrogate the ability of PBMCs from these HIV-infected individuals to make a specific T-cell response. And we found in HIV-negative people, as expected, very low background levels to HIV peptides, very limited HERV peptide recognition. But when we looked at HIV-positive people, we saw, in many cases, responses to endogenous retrovirus peptides. If you take one particular individual as an example, this is patient OP581, they make individual peptide responses not only to the HIV peptides, but also to endogenous retrovirus peptides. In some cases, at even greater magnitude than to the HIV peptides. So this endogenous retrovirus response had not been recognized as contributing to antiretroviral immunity until then. So as we've already heard, it's from Hendrik, it's difficult to make studies in humans that develop into mechanistic studies, but from a correlative perspective, we found that there was an inverse relationship between the plasma viral load and the number of HERV-specific T cells. So then, was that something only found in primary infected patients? So what about chronic infection? So the hypothesis here is that controllers, elite controllers, would have HERV responses of higher magnitude or breadth than other individuals based on their inverse correlation from primary infection. So Devi Sengupta in my lab, along with Ravi Tandon and our colleagues in Toronto, asked the question whether there was any difference in the HERV-specific immune response in people with chronic HIV infection. And again, she used an ELISpot assay to look for interferon gamma production, and then also looked at individual patient studies. So she had a number of different groups, 22 HIV-negative people, immunological progressors, viral non-controllers, people who are heart suppressed, and elite controllers of a similar age and reasonably similar ethnicity. And what she found, and we've recently published this in the Journal of Virology, is that the elite controllers were those that had the responses at higher magnitude. Again, supportive of the concept that having this autoimmune HERV response is not deleterious in HIV elite controllers. She also did what Keith and Brad had done and found that there was an inverse correlation within the chronic group between viral load and HERV response. So HIV-1 controllers have these stronger HERV-specific T-cell response, and it's inversely correlated with HIV viral load. So a few years ago, we had been following in the literature a really interesting study from the cancer immunologists. And Richard Childs at the National Cancer Institute had published a fascinating paper in which he was following people who had renal cell cancer. And they had had an allogeneic stem cell transplantation and had cleared their metastases. And he discovered that the regression of the human renal cell cancer was related to T-cell responses against HERV, a different HERV family. And so what they did, or asked the question, is could that donor immune response from the transplant be directed against, in this case they were thinking of tumor-restricted antigens, antigens which were restricted to the cancer cells, and could they identify that? And by a clever reverse immunogenetic method, they could look using ELISpot at people who had had these donor cells, and they could identify the tumor antigen as coming from HERV. So this we thought was pretty exciting because this suggested that in vivo, in people, HERV-specific T-cell responses might actually do something. But how can you then take that one more stage further and ask what's happening in HIV infection? So Brad Jones in Mario's lab set up a series of heroic experimentations to ask the question, could there be recognition or suppression of HIV-infected targets from cloned HERV-specific T-cells? And in order to do this, he cloned HERV-K envelope-specific cells, and from the same patient HIV, GAG, and CMV specific cells, and then co-cultured them together with a number of different target cells that would ask the question, was the recognition of HIV-infected cells? And the CMV clone tested in this co-culture system looking for interferon gamma production, or CD107, as you've heard from Hendrik's talk. There was no effect. You would not expect a CMV clone to recognize and kill an HIV-infected cell. The HIV GAG clone did nicely recognize and cause degranulation, shown there. And so did the HERV specific cells, in this case tested on a peptide or 3 different primary isolates, because if this concept is to be validated, then we would need to be able to show that all the clades that we could test independent of HIV sequence would be recognized by the HERV-specific clone. And they were. So the model stands where we would be able to elicit HERV-K or HERV of certain specificity-specific T cells, and they could recognize HERV expression and eliminate an HIV-infected cell. So that is the concept which we hope will go through to be tested further. So to summarize, HIV and HERVs share similar epitopes as well as have unique epitopes, and these HERV-specific cells are capable of recognizing HIV-infected targets. So we think that this is important in understanding not only the natural control of HIV infection, but also could provide a new target for vaccines and immunotherapeutics. And one of the advantages is that these are stable and lack of variability. Of course, what we're considering here is eliciting, if you like, an autoimmune reaction. But it's a different autoimmune reaction in the sense that we believe that HERV-K is expressed only within an HIV-infected cell, and that's what we want to get rid of. And the elite controllers are walking around without obvious deleterious effects from autoimmunity. As we progress in this study, there are quite a few further processes that we need to go through. So in the last few minutes, I'm going to turn to the next part, which again is related to HIV and autoimmunity. And this is to show you the data that people who have the skin disease psoriasis have a set of genes which make them susceptible to psoriasis, which are extremely similar to those found in HIV elite controllers. And working with a dermatologist in San Francisco, there has been a puzzling clinical observation in people with psoriasis that HIV infection actually makes psoriasis worse. And treatment of HIV improves psoriasis. And people actually don't know or haven't known why this might occur. But we thought that there may be a clue from the literature. And one of the first things that we started to look at when that famous science paper came out that you've seen several times during the course showed, again, that HLA region is important in HIV elite control. The psoriasis geneticists have been looking with GWAS studies as well and finding certain associations. Who are HIV elite controllers? Well, Dave Watkins has already taught you about this. These are people who are a rare population who are infected with HIV and have naturally good control of viral replication without antiretroviral therapy. And there are some people who have been living with HIV for around 30 years without being treated who are doing well. So the hypothesis that we came up with is that psoriasis is a disease that's marked by overactivation of a set of antiviral genes that under normal circumstances help humans fight retroviral infections. And if that was true, then we would be able to say that gene variants that confer antiviral immunity should be enriched in people who have the skin disease psoriasis. So in thinking about the mechanisms that we know are important for HIV elite control, we know there's peptide binding by certain HLA-B alleles, increased HLA-C cell surface expression, and also natural killer cells, along with other important criteria that help build an HIV elite controller haplotype. So we know that the HLA class 1 alleles, B57 and 27, are protective against the development of AIDS. Here, B57, if people have B57, they do very much better. If people have B27, they also do better. But there are also unfavorable B alleles. So the B35PX allele actually means people do much worse if they're HIV infected. So there's a range. There's not only an HLA association with control, there's also an HLA association with progression. And the study from the Walker lab and others have shown the importance of the HLA-B peptide binding residues, and you'll recognize from Ken Rock's talk the hot dog bun with the sausage in it, and the important residues that are associated with binding peptides 97, 67, and 70 are residues that Bruce Walker GWAS study identified as being absolutely critical in controlling HIV. Mary Carrington's group and others have shown, again for HIV control, that there's high levels of HLA-C expression, and the higher the level of HLA-C expression, the lower the level of virus, and that's independent of HLA-B57 and 27. And there's an important role for natural killer cells here, and there's an epistatic interaction between the HLA-B alleles, and, or some of them, and KIR3DS1, that are also important in delaying progression to AIDS. So we know from the HIV elite controlling literature there's some good HLA alleles, some bad HLA alleles, and that these are mediated mechanistically by certain amino acid positions within that groove. So when you look at psoriasis patients, at a number of different cohorts that Wilson Liu, who is my collaborator in this, has studied, has looked at a large number of different people with psoriasis, compared to a large number of controls. And what he did was he did a technique of HLA imputation that allows you an integrated framework to impute classical HLA alleles from SNP genotypes. So that we didn't have to go back and HLA type all of those individuals. And this imputation method allows you to identify people of certain HLA types. And there was really good concordance in this method with a 4-digit resolution. And so we felt that this imputation method was valid to then ask the question, within the psoriatic genes, what is the representation of those HLA alleles? And to our surprise, we found that in people with psoriasis or HIV elite controllers, that the p-values for these HLA alleles were really extremely similar between people with psoriasis and people with HIV. And these were adjusted for gender, ancestry, and cohort. And then Wilson did a stepwise regression model to identify independent effects. And independently, B57 and 27 were really important in protecting or susceptibility in psoriasis. And interestingly, B35 significantly protects against psoriasis. So B57 and 27 make you susceptible to psoriasis and good at HIV control. Having B35 protects against psoriasis, but is bad for HIV control. And then taking the Bruce Walker model and asking the question, can you look at each of those amino acids that were important for binding? Again, the top 5 amino acid positions for psoriasis occurred at the 3 positions, 97, 67, and 70, which were absolutely essential for HIV control. And the p-values for psoriasis here are extremely strong. And it may be hard to see at the back here, but the top panel, psoriasis, this is HIV, and then looking at other autoimmune diseases, this is rheumatoid arthritis, this is Crohn's disease, this is type 2 diabetes, this is coronary artery disease. We didn't see the same imputation. There is some similarity with type 1 diabetes, but the big link here is that the HLA-B amino acid positions for psoriasis and HIV elite control are the same. We then went on to look at the HLA-C region, and Mary Carrington had another paper recently in Nature where she showed that there was a polymorphism in the 3' UTR of HLA-C, which is enriched in HIV elite controllers. Again, there it is, really strongly enriched in people with psoriasis. NK cells, again, psoriasis, same combination as is found in HIV elite controllers. So when you look at HIV elite controllers and people who are susceptible to psoriasis, there are these similarities. There's increased B57-27, decreased B35, these important amino acid residues, high HLA-C expression, and NK cell activation. So we think by this study of population genetics that we have been able to identify a new concept that will allow us to go on to dissect potential antiviral mechanisms. And we know that there are a set of viral control genes that are protecting against HIV. They are the same viral control genes that are leading to an increased risk of psoriasis. And so we think we can now explain the psoriasis HIV-1 paradox. In either a psoriasis patient or someone with HIV, you get an intense triggering of a response through these mechanisms, which leads to an inflammatory cytokine milieu, which allows the psoriasis to flare. If you treat people with HIV who also have psoriasis, then you reduce the immune response and the psoriasis improves. But this psoriasis area is taking us down a very interesting route of trying to dissect out antiretroviral mechanisms. And we also have a paper in press that actually ties in a potential antigen from endogenous retroviruses in people with psoriasis. And that may be the trigger that's occurring in this circumstance. So I'd like to stop there. It's been an enormous pleasure to work with colleagues, science is a collaborative program, and with Mario Ostrowski at the University of Toronto and his group, particularly a really amazing grad student at the time, now postdoc at Harvard, Brad Jones. And within my own lab, we had a number of wonderful people working on it. Lish is here. Rafaela, I don't know, she may not be able to be here, but she was a medical student who came to my lab for a year and really contributed an enormous amount. And Keith Gowson, a postdoc who's now a professor, really worked closely with Brad, and Wilson working on the psoriasis. And we've had some really good interactions here with Esper Calas and his group, and from the psoriasis perspective, Mariana Bautista in particular. So thanks very much indeed.